Should we start, guys? Okay, so we're continuing from last time, where we talked about DNA and mentioned in passing RNA, and the genetic information is from DNA to RNA into protein. What I've done on this slide is I've arranged so you can see the differences between RNA and DNA and the main differences that you would be expected to recall and apply. So the key difference with the sugar is that in DNA you have deoxyribose, and that's what the D stands for, whereas in RNA it will be ribose, so that's why it's R for RNA. And in terms of nucleobases, RNA has uracil, whereas DNA has thymine, and the difference between those two is just one methyl. So in uracil, there's no methyl, whereas in thymine, there is a methyl. So this allows uracil, the lack of the methyl, allows RNA to form actually very much. It allows the RNA to bend a lot more, so you can get lots of different types of secondary structures. So in terms of the secondary structure, DNA is double-stranded, whereas RNA will be single-stranded and can fold upon itself. So it's a single strand, but it can have secondary structure, but that's involved by one strand folding upon itself. So the Chargaff rule does not apply to RNA. So you don't get the same number of A and U, for example, as you would for DNA, whereas A, the amount of A you'd get, you'd always get the same amount of T. DNA is always longer. So DNA is a very, very long molecule, whereas RNA can be between 75 and a few thousand nucleotides, but it's not going to be as long as DNA in the length. So that's kind of the main differences between DNA and RNA. So a question you could get in an exam might be compare and contrast RNA and DNA. So what's the same, what's different? Could you identify the bits that are the same? Could you identify the bits that are different? So that could be a nice exam question for an example. Are there any questions on differences between DNA and RNA? Okay. So RNA, as I mentioned, can form additional secondary structures. So you can have double helical RNA, and if you do, it's in the A form, and the A form is more symmetrical than B form. So the major and the minor grooves are roughly about the same size. So the minor groove is shallow, and then the major groove is quite deep. But the key thing with RNA is you can form lots of different secondary structures. So this is an example of tRNA, and that's what you usually see it depicted as in a textbook. And this is showing that the RNA, it's one single strand that folds upon itself, and then you can have base pairing within that. So tRNA is important in translation, and then we describe it as having a cloverleaf shape because it has these different hairpins, and then these are kind of loops, so these kind of bulge out. So this is where there isn't any base pairing in there. And the flexibility of RNA is derived because of the difference in the sugar, but then also the fact that you've got uracil instead of thymine. That lack of the methyl means that it can adopt more complex shapes. So RNA is less stable than DNA, and then it goes through a hydrolysis mechanism. So here's a general mechanism for that hydrolysis. So we've got an OH, which is representing our base, that can take off this proton from the 2' hydroxyl group, and the electrons from there would then go on that oxygen. These can then do an so So that means within the same molecule, intramolecular attack on this phosphate. So this negative charge goes to here, and then these electrons go on to this oxygen here to give you this intermediate. These electrons will come back and then break this bond here. So what happens is you get cleavage of the strand. So under basic conditions, RNA will actually break down. So it's much less stable than DNA. So I'd expect you to be able to draw this mechanism. If we gave you a segment of RNA, it's a hydrolysis mechanism, so it's relatively similar to the ones that you've done with Dr. McDermott and Professor Searcy. So what conditions do you think could make this happen quicker if we were doing hydrolysis? Any ideas? Yeah?
Water in acidic conditions, but alkaline conditions are first and then acidic conditions.
So if it's going to happen more likely in alkaline conditions, so if you want to stop it from happening, you could make it a little bit more acidic, so reduce the pH. If you wanted to increase that reaction, making it a bit more basic would help. So pH, you could change the pH. What else? In general, how do you make a reaction go faster? You can heat it. So in humans, our physiological temperature is 37 degrees. If we were working on RNA in the lab, we'd actually put it on ice to make sure that any potential hydrolysis reactions didn't happen as quick. So we could have more time with the RNA before it self-destructed. So why is this important? So RNA is less stable than DNA. Why? There's a reason why. Yeah?
So it can assemble and disintegrate quickly in different processes such as transcription.
Yes, so it can disintegrate quickly for things like transcription. So your DNA is your genetic code, that's your blueprint, and you need that to be stable over a period of time, for at least the lifetime of that organism. Whereas RNA is transient, so you use the RNA as a working copy. So you make a little copy of the bit of the DNA that you need, and for example, you might use that to make a protein, but you might not want that protein all of the time, so that's the reason why RNA is actually less stable, so it gets broken down. So if you need that protein again, you need to make that RNA again, and that's how it works. Rather than just having RNA hanging around all of the time, this is actually a way of deciding how much of a particular gene we need. Any questions on that? Okay. So this is the flow of genetic information. So we've got our DNA, and that can replicate. So we call that replication. So this is when, if you're making new cells, if you're regenerating things, the DNA needs to be replicated for that to happen. And that gets converted into RNA. And RNA is the working copy. And that RNA can then get made into proteins. So there's lots of different types of RNA. So you can have messenger RNA, which gets converted into protein. RNA can be used for RNazymes. So these are enzymes made out of nucleic acids that help reactions progress. And then here's the example of tRNA, which helps in translation. And then that makes protein. And then each of these different parts of the flow of genetic information has an associated sort of name. So anything to do with DNA is called genetics or genomics. Everything to do with RNA is to do with gene expression profiling. And then everything to do with proteins, proteomics. So there'll be people that specialise in looking at all of the different proteins that to do with different organisms. So why do only certain organisms have certain proteins? Why do we have code for proteins that we don't use, and things like that? So before a cell can divide, it has to replicate all of its DNA. And to do that, it will take the two strands apart and then use each one as a template to duplicate the DNA itself. And this process is called replication, and that process, by taking the DNA apart and then using it as a template, we call a templated process. So it's quite intuitive. And because of the way that it is our blueprint, it needs to be extremely accurate. So DNA needs to be stable, but the replication of DNA needs to be accurate, because if you don't get the code right, then you're going to introduce different things, the code for different things that you don't want. And the human genome's 3 billion base pairs long, so it is a very, very complicated task as well. And what happens is you'll have your template strand in your DNA, and so this is thymine, and this is base pairing with adenine, this is cytosine base pairing with guanine, and this is adenine base pairing with thymine. And what happens is your template strand's already there, and then you have a growing DNA strand. So this is the growing DNA strand. This is the new base that is being introduced, and notice that it has a triphosphate here. So this is actually deoxyribonucleotide triphosphate. So this is the thymine equivalent of that. And you probably, if you did A-level biology, or you probably already learnt about ATP, adenosine triphosphate. This is a provider of energy source in the cell. And so if this was the adenine, this would be the adenosine triphosphate instead of the thymine triphosphate. So obviously this is a source of energy, but this will then interact with this, react with this to actually cause this phosphate diester. So they do it one at a time, they template, so they base pair first, and then that reaction will occur between these two bases. And this is how you create the polynucleotide chain. And as we mentioned before, the DNA has a direction, and so the growing chain is from the 5' to the 3', so you're always adding on to the 3'. So you work from 5' to 3'. Are there any questions on that? You'll need that for your pharmacology modules to understand about DNA replication. So we're going to just briefly go over DNA as a therapeutic target, because you see that's probably what a lot of you are interested in. So the main target and the main disease that you target DNA for is typically cancer. There are other diseases, but the majority of the time, most of the time, it will be cancer. And there are many different cancer treatments, but the ones that target DNA are often targeting replication of DNA. So cancer is a disease where you get uncontrollable cell replication. So your cells are dividing rapidly and rapidly, so what can you do to stop that? And quite a lot of these will target DNA directly or indirectly. So by directly, they could be alkylating agents, or things like cisplatin or natural products, or they could be indirectly. So you're targeting the enzymes that process the DNA, so the things that actually help DNA replicate. So in terms of targeting DNA indirectly, so this is a diagram of a replication fork. So you'd have your DNA polymerase running along this, this single strand, and it's replicating that piece of DNA. You've got helicase, which is an enzyme which helps unwind the DNA, and then you've got an enzyme called topoisomerase that actually can cut the DNA, but it actually relieves some of the tension. So last time we talked about actually how DNA gets wrapped up very, very tightly around histone proteins. When you start to unravel it, to either replicate it or even to read it, some of that helical tension needs to be relieved, and that's what topoisomerase does. So there are certain examples of, we would call these DNA processing enzymes, so the helicase, the polymerase, the topoisomerase, they all help that DNA to do what it needs to do. There are other There are some compounds that will target these processing enzymes, like topoisomerase. So etoposide, that's an example of one of those compounds that targets topoisomerase. And amzocrine, which is an acridine derivative, also targets topoisomerase. But you can also have what are known as antimetabolites, and these are compounds that look like a DNA or RNA base, and they can get incorporated into the growing strand, but because they're not quite the same structure, that will then cause a difference in their metabolism. So that's why they're called antimetabolites. So examples of this is acyclovir. So this is an antiviral. I think it's marketed under Zavarex, but it's usually used for cold sores. And this is AZT. So, and the AZ comes from this group here, and it's a thymine. So this is an azide functional group, and AZT is used to treat HIV. So these are kind of antivirals. So these will actually stop the virus from replicating by mimicking the bases that virus needs to actually create another virus. You can also target DNA directly, and there are many different ways you can do that. And one of them is through intercalation, which is when you have a molecule which slots in between the bases. So if you think about the bases a bit like if you think of them a bit like a spiral staircase, and you've got your steps going up, that space between those steps, you can have a molecule slot in. And that's what we call intercalation. So if a molecule is going to do that, it needs to be flat. So molecules that intercalate have flat planar surfaces. So chloroquine has a very flat planar surface there. Doxorubicin has the same flat planar aromatic surface where they can slot in there. Another way molecules can interact is by electrostatic interactions with the sugar phosphates. So quite a lot of these compounds you'll find, so the sugar phosphate backbone is negatively charged. So quite a lot of DNA binding compounds have positive charges on them. So chloroquine here, that would be positively charged at physiological pH, and doxorubicin here has an amine that would be positively charged at physiological pH. So these add extra interactions, so they can bind somewhere in the DNA, but then they have what we call a side chain that can interact with the sugar phosphate backbone. So it's like an arm that would grab round and get an extra interaction. So these would then bind using two different types of interactions. You can get compounds that alkylate DNA, so that means that they might cross-link DNA, or they might actually add a functional group to DNA. So cisplatin is one of those, and this is very, very commonly used in a lot of cancers. You can target the minor groove. So here, this is the major groove, so the distance between here and here is larger than here and here. So this is your major groove running along here, and then this is your minor groove because it's smaller. So in the minor groove, you can get molecules like distomycin bind. So distomycin, as you notice, it's curved, and that curve follows the curve of the DNA. So proteins will usually interact in the major groove, but actually in the minor groove it's small enough so you can get small molecules interact in there. And then the last example is that you can have compounds that will cut the DNA, and that's called DNA cleavage. So bleomycin is a natural product that has a very, very reactive iron species, which will then create radicals that can cleave the DNA. And then your body will recognise where there's bits of DNA that aren't connected together, and they can then go through a repair process. So I'm going to give you a case study of a type of cross-linking agent called nitrogen mustards. And these are actually derived from sulphur mustards. So sulphur mustards were used as mustard gas in the First World War. And people would breathe that in, and actually it would actually kind of almost liquefy your lungs. But they knew that there were different things that these compounds would cause, and they would cause a lot of cancers. And that seems like a bit counterintuitive, but actually what they tried to do in the 1930s was see whether we could use these compounds in tumours for humans. But the problem is these compounds are sulphur mustards. They're very, very reactive, and then that means you can't use them systemically, so you can't introduce them to the body without them actually causing more damage than they would help. And this is an example of a nitrogen mustard, and these were derived in the Second World War as a chemical agent, again for warfare, but these were investigated in humans during the 1930s and 40s for their effects on humans. And some of these were developed into drugs. So here's some examples of nitrogen mustard drugs. And their structures are slightly different to the mustard and sulphur mustard gases. And the reason being is through their mechanism of action. So if I explain how these work, you'll kind of soon see why some of these are more reactive than others. So this is an aniline mustard, and it's called an aniline because it's if you remove these two groups here, you'd be left with an aniline. This is melphalan, which is also known as LPAM, and it's actually an amino acid. So if you look here, there's an NH2, and there's your carboxylic acid, and this is an amino acid. And then chlorambucil has a carboxylic acid group here as well, so that affects how that molecule actually interacts in the body. So nitrogen mustards cause crosslinks between DNA. So what they do is they do inter-strands. So if you've got two strands of DNA, you'll cross-link the two. When that happens, if you imagine you're an enzyme, you're trying to pull apart that DNA to replicate it or to read it. And if you can't pull it apart because it's got cross-links, that's going to affect your ability to for cells to divide. So it's going to be able to stop cell division. So it will then stop replication and stop cell division. And they predominantly cross-link on the N7 of guanine. So this is our GC base pair here, and this is the N7 of guanine. So the mechanism of alkylation is very similar to an SN2 type substitution. So the lone pair on the nitrogen will attack this electropositive carbon, which is next to the electronegative nitrogen. So you can draw an arrow from there to there, and then the electrons from this bond will then go on to the chlorine to give you a chloride. And this forms a very, very strained cyclic ring, which we call an aziridinium ion. And this is very reactive. So this happens very readily because chlorine's a good leaving group, chloride's a good leaving group. And then this forms this reactive species, the aziridinium ion, and this allows then, typically, the N7 of guanine. So this is guanine, this is the nitrogen 7. The lone pair can then attack in here, relieve that strain, because it's a very strained ring, and then neutralise that positive charge to give you this. And that gives you a monoalkylation adduct. But the key thing with this is, you've still got another arm of this molecule to do exactly the same mechanism again. So this is how the molecule cross-links the DNA. So the molecule bridges that gap. So you see the exact same mechanism happen again. The nitrogen lone pair attacks that electropositive carbon, kicks off the chloride, and then forms the aziridinium ion. And then you can have another guanine, and the lone pair will then attack here, and then relieve that strain there to give you a GG crosslink. So that molecule is essentially bridging those two guanines together. So that's what we mean by a cross-linking agent. And if you were to draw this in an exam, if you're given the structure of guanine, brilliant, but you could just write G with a lone pair, that would be fine. So the reactivity of this depends very much on what is attached to that nitrogen, and it's all about the availability of that lone pair. So is that lone pair available to attack here in the chlorine, kick off the chlorine? If you've got an electron withdrawing group here, that's going to withdraw some of the electron density away from that lone pair, and that's going to result in reduced reactivity. Whereas if you have an electron donating group, that's going to make that more reactive. So if you have an electron donating group, such as a methoxy on an aromatic ring, or something that can donate by the inductive effect, such as a methyl group, that's going to donate electrons, and then that's going to make this more reactive, because if you've got more electron density here, this lone pair is more available, and then that's more likely to happen. Whereas if you have an electron withdrawing group, the exact opposite is going to happen. So if you had a CF3 group or a nitro group in this R position, all of those electrons are going to be not available so much to attack because they're being sucked back in by these electron withdrawing groups. So this would result in reduced reactivity for these types of compounds. So it's the formation of this aziridinium ion which means that you get different reactivity with these nitrogen mustards. So for example here, when you have different substituents here, that's going to affect whether this lone pair is available or not. So in aniline mustards, compared to the original compounds that just had a methyl group, when you just have a methyl group, you're going to have sigma donation towards that lone pair, and therefore that's going to be very reactive. But if you have an electron withdrawing ring, these electrons can then donate into the ring, and actually they are delocalized into the ring, so they're less available. So this means that these compounds aren't as reactive, and that means that they're less toxic. So you've got this better balance of something doing what you want without being too toxic for use. And just to be clear, when we talk about intra versus inter strands, this is what we mean. So an intra strand means between the same strand, like this, whereas an inter strand means that you're cross-linking two strands of DNA. So it's the inter strand which is correlated with the anti-tumour activity, because you're preventing the DNA from being able to pull apart. So here's a summary of the different types of alkylation that you can get. So we've talked about the form with the aziridinium ion, and it's like an SN2 type mechanism. So it's an intramolecular reaction where you've got your lone pair attacking into the electropositive carbon. These bonds break and go onto the chlorine to give chloride. You get your aziridinium ion, and look, I've just put DNA G with a lone pair to simplify it there. That attacks here, and then these electrons go onto this positive charge, so you get this DNA adduct, and that can happen for both these. But you can also get direct substitution by SN2. That is also possible. It's less likely because this happens so much more readily, because intramolecular reactions always happen quicker than intermolecular reactions. So if you've got a molecule that can fold upon itself or react with itself, it doesn't have to find another species because it's all there already, all ready to go. Whereas when you have two species that got come together, that's going to always be a little bit slower. So that's why intramolecular reactions always happen faster than intermolecular reactions. So I'm just going to cover a little bit about some research that goes on here at UEA, because it's important that you have an appreciation of different types of things that go on. So Professor Searcy works on a lot of compounds that are sort of usually derived from natural products, and one of those are the duocarmycins, and these are very, very potent anti-tumour antibiotics. And the reason they work very, very well is because they work by an alkylation mechanism. So notice this functional group looks very similar to that of the nitrogen mustards, but they will only react, these act like proteins, they only react when they bind DNA. So this molecule actually lies slightly out of plane, and only when it binds to DNA will it be flat, and then it's then really, really, really potently reactive. So these are really, really potent compounds, and actually in the CSE group what they're doing is they're conjugating these with antibodies to try and make them very specific for certain types of cells. So antibodies can recognise cells, and in particular you can get antibodies that recognise cancer cells over normal cells. So they're trying to combine the two, combine the very, very potent drug with the antibody to help get improve the specificity of those compounds. And in my group, we do a lot of work on alternative DNA secondary structures. So this is like a Watson and Crick double helix, but then we work on four-stranded structures, like G quadruplexes and I motifs. So these are very, very tightly compact structures. And as you can see, they look very different to double helical DNA. So we're trying to make compounds that drug-like compounds that combine these, so we can see what effects they will have on gene expression and also in the development of genetic diseases. So we've actually found, or we work on several different types of structures, but I'm very interested in working on one that's involved in the insulin gene. And what we've found is that the insulin gene varies between different people, and our hypothesis is that dependent on the sequence you'll get different versions of these structures form, and we can potentially use those to predict whether somebody is likely to develop diabetes or not. So it's not always about cancer when you're talking about DNA either. So there's some research that we've done relatively recently that has involved DNA, and some of that kind of has been done in collaboration with other people. So here's an example of some work that I was doing in collaboration with somebody in the School of Biology, and we made these compounds, these two here, and they interact with this special type of DNA, and we were able to switch off a gene pathway in bacteria, specifically using these compounds. And so that's an example of research that actually happened here at UEA. And the reason although this is a slightly older article, the reason that I keep this one on here is because Bavina and Tim Meadows were undergraduate students that worked on this project. So their work that they did was published in a peer-reviewed journal, and that will probably be one of the papers that I include for my research assessment. So some other compounds that can be made or might be interesting to you is, this is another example from a paper showing that you can have different structures of compounds. So these are like nitrogen mustards, so these are different derivatives of these. And some other examples of different compounds that bind, so these are ruthenium complexes that can actually switch on. So they fluoresce, they light up when they bind DNA. So I think with the version of the lectures that I uploaded, you might have had some different research hot off the press, but if anybody's got any questions about that, just come and let me know. But I kind of wanted to just go over the fact that DNA and RNA are different. Remember that RNA contains uracil, which doesn't have that methyl group on, not thymine. It's less stable, and it's usually going to be single-stranded, so it's not going to adopt a B-form structure. Flow of genetic information, DNA, RNA, protein. We used to call it the central dogma, but we like to challenge dogma nowadays, so we don't tend to refer to it as the central dogma, but you will see that in textbooks. DNA replication is templated, so you use one strand to make the other. So A base pairs with T, G base pairs with C, so you always get the exact opposite of what you've already got, so you can always make a perfect copy. And you can target DNA using the major groove, minor groove, the backbone, you can do alkylation, you can do cleavage, and you can target it indirectly by targeting the processing enzymes and the way that DNA is synthesised. And then we looked at the nitrogen mustard reactivity and properties. So if anybody wants to do any more reading, there's plenty of places to look. So Jurek has some really good stuff about organic chemistry. Cell biology, the Albert's book is really good. And then I've got some links to some websites that actually have really, really good information as well. So if anybody has any questions, I'd be prepared to answer them. If not, I guess I'll see you guys next year. This is my last lecture for this year, so don't be sad. Hopefully,